Okay, so let's get started with this test review. This is going to be the first of a couple of videos. You'll see I've got my review all stuck into my notebook, and I've got my anaerobic card taped in. I'm going to scoot that out of the way, and we're going to start here. So the question says, photosynthesis where? Which are the organelles which are directly related with the production of protein synthesis? So, I know that transcription starts in the nucleus with DNA being converted into messenger RNA. So I know the nucleus is part of one of the organelles, or it is one of the organelles. And I've got the nuclear membrane there. I know that once the messenger RNA has been made, it goes to these little organelles here that are attached to the endoplasmic reticulum, where translation happens, and they are called ribosomes. So in ribosomes, the messenger RNA is used to assemble amino acids in a specific order in the protein. Once the amino acid chain has been produced, it is then taken to the Golgi body, where it's modified, things can be added, it's folded into specific shapes, and at that point it is transported to wherever it's needed, so that we have the Golgi body. So what are the three main organelles involved in the synthesis of proteins? The nucleus, ribosomes, and the Golgi body. Okay, moving along to these two. Clearly, we've seen these plenty of times before. These are the building blocks of nucleic acids. These are called nucleotides. So, whoops. Okay. And since the two nucleic acids that we know about are DNA and RNA, we need to know what they're like. So both of them have a phosphate, the P part, and they both have a sugar, and the sugar is in so the sugar in DNA is why DNA is called DNA. The sugar is deoxyribose. OSE always tells you it's a sugar. So in DNA, the sugar is deoxyribose. In RNA, the reason it's RNA and not DNA is because the sugar is just ribose. R-I-B-O-S-E. So that's sort of difference between the two nucleotides. Now, the third part of a nucleotide is the nitrogen base, N base. And we know that in DNA, the nitrogen bases are A, T, G, and C. And in RNA, the nitrogen bases are A, U, G, and C. That's a way we can determine whether it's DNA or RNA. Is there a T, thymine, or is there a U, uracil? So, carrying on from that, how do you distinguish? Remember, the term distinguish means tell apart. How do you tell them apart? Okay? And it's really simple. You have to look for what their differences are. So how do you tell an A base in DNA and an A base in RNA nucleotide? It's going to be by the type of sugar. But in essence, if you're just looking at the DNA nucleotides, you tell them apart by the nitrogen bases. So how do you distinguish between nucleotides? The identity of the nitrogen base. Is it adenine, thymine, guanine, cytosine? Or if it's RNA, is it uracil? Okay. This one here, this is kind of a bit of a throwback to a previous unit. Complementary base pairing in DNA. So if this is one side of DNA and you're asked to complementary base pair it with the other side, you're going to say, what would, if there's an A on this side of the DNA, what would be on the other side of the DNA? And so if there's an A on one side, obviously it's going to be T. If there's a G, it's going to be a C. If it's a C, it's going to be a G. And if it's a T, it's going to be an A. And it might be a little easier if you looked at it this way. You can now see you've got both sides of the DNA like that. And then you would have, you remember the hydrogen bonding that kind of go in between each one. So if you have the complementary base pair DNA, one side to the other, it's just A goes with T, G goes with C. Okay. All right, looking at these two things, we have two sections of nucleic acids. I'm looking at this one. I see a T. I know immediately that this is DNA. No problem. I see a U in this one. I know immediately it is RNA, because you only have T's in DNA and you only have U's in RNA. So the next thing is, what do you call 3 nucleotides or 3 nitrogen bases in DNA? That is a gene. All right. And what do you call 3 nucleotides or 3 nitrogen bases in RNA? That is a codon. And when we're trying to work out which amino acid is being coded for, we use a codon chart. We do not use a gene chart. The codon chart tells me that if I'm given a gene, if I'm given 3 bases in DNA, the first thing I need to do is convert it into RNA so that I have codons, and I can go to my codon chart and I can work out which one's which. Okay? And while we're talking about codon charts, and so we're not having any confusion, let me just pull it up here in my book. Right? This is what we're talking about when we're talking about codon charts. If you see a codon chart with a question in a test, it means you have to use the codon chart to answer the question. If you do not need to use a codon chart, it will not be there. But if it is there, you do definitely need to use it. Okay, let's scoot on to this bit. Okay, just a little more vocabulary. SERLEUARGMET, serine, leucine, arginine, methionine. What are they? They are amino acids. Okay? And amino acids join together with bonds to form proteins. There's another word that we use for amino acids joined together in this form. It is polypeptide. So a polypeptide chain is the first level of organization of a protein. Okay, moving down here, this is something we already covered in this little card, but just a quick refresh. Aerobic respiration, we know that aerobic means with oxygen. Okay, so what are the reactants for aerobic respiration? Well, the initial source of energy is glucose. And obviously, because it's with oxygen, we need oxygen. What do we produce during aerobic respiration? What are the products? We exhale carbon dioxide, which is a waste product, and we exhale water in the form of vapor. But the key bit here is, why do we do aerobic respiration? Because we're taking the energy stored in the bonds of the glucose and we are converting them into 36 units of ATP. ATP is the form of energy that cells use. Now, coming down here to anaerobic respiration, ANA, if you put that in front of a word, it means the opposite. So it's the opposite of aerobic, so it's without oxygen. Okay? And so, because we don't have the oxygen that we had before, the glucose is broken down, and it's broken down into an intermediate product that builds up in your muscles called lactic acid, or sometimes it's called lactate. Okay? Now the big deal here is that anaerobic respiration is a lot less efficient than aerobic. So for every 1 molecule of glucose, you only get 2 lots of ATP. This is, as I was saying to you before, this is what we do normally, which is much more efficient. I would much rather get 36 lots of ATP from 1 lot of glucose. But in the event that I can't get the oxygen to my cells when they need it as fast as they need it, I have no choice. Those cells need ATP. We're going to start breaking it down without the oxygen, and we're only going to produce 2 ATP. But 2 ATP is better than nothing. Okay, I'm going to stop this video here. This is going to be the end of the first part of the test review.